Students, in this video I'll be discussing about the structure of nephron and process of urine formation. Now we have seen that the main filtering organ in our body is the kidney. So there must be something in the kidney which will filter the blood to take out or remove the nitrogenous waste. So the filtering unit in the kidney is the nephron. So this is how a nephron looks like. Nephrons are very minute structures. In our kidneys there are around 1 million nephrons. So the nephron first begins with a hollow cup-like structure, and this hollow cup-like structure is called Bowman's capsule. From the Bowman's capsule, a tube arises. The initial part is highly folded. It is called PCT. Full form of PCT is proximal convoluted tubule. Tubule means tube, convoluted means folded, proximal means it is near to the Bowman's capsule. PCT then leads into a hairpin-like bend which is called the loop of Henle. After that, it forms another coiled structure which is called the DCT. DCT stands for distal convoluted tubule. Distal means away from the Bowman's capsule. The DCT then goes and joins the collecting duct or the collecting tubule. Now, blood has to be supplied to the nephron because nephron will be filtering the blood. So blood is brought in, unfiltered blood. So we should not make the confusion between deoxygenated, oxygenated blood and filtered and unfiltered blood. So deoxygenated and oxygenated is in relation to presence or absence of oxygen. Filtered and unfiltered blood is in relation to presence and or absence of your urea or nitrogenous waste. So the blood will be brought in by the renal artery. Each kidney receives one renal artery. So the renal arteries will branch out. A fine branch from the renal artery will come and it will enter the hollow space of the Bowman's capsule. After entering, now this incoming blood vessel is called the efferent arterial. Efferent means to bring to. So it is bringing the unfiltered blood into the Bowman's capsule. After entering the hollow space of Bowman's capsule, it divides and forms a huge network of blood capillaries. And this network, this ball of blood capillaries is called glomerulus. Now from the glomerulus, the blood capillaries will join and they will come out again as an arteriole which is called the efferent arterial. The efferent arterial will go, it will coil around the PCT, DCT and the loop of Henle. And from midpoint it is again known as venule, and this will go and join the renal vein. So this is the overall structure of nephron and how blood is supplied to the nephron for filtration. Now let us have a look at urine formation. Urine formation in the nephron can be divided into 3 steps. Number 1, it is called ultrafiltration. Ultrafiltration means filtration under very high pressure. So basically when unfiltered blood is coming out, from the blood the fluid component of the blood will come out, which will include mostly water, urea, then salts. All these components will be present in the fluid which will be coming out from the blood and entering into the Bowman's capsule. Now that fluid is called glomerular filtrate. So since the diameter of the capillaries is very fine, therefore the pressure of blood is very high. So the filtration which is occurring here is occurring under high pressure. Therefore it is called ultrafiltration. So upon ultrafiltration, a lot of fluid will come out from the blood, and the thick blood which will contain little bit of plasma, blood cells and big size proteins, they will keep on flowing and it will come out through the efferent arteriole. So this glomerular filtrate which comes and enters into the Bowman's capsule, it then starts flowing through the PCT, loop of Henle and DCT. By the time the glomerular filtrate will flow through the PCT, loop of Henle and DCT, most of the essential components present in the Bowman's capsule will be absorbed back. So what are the essential components present in the Bowman's capsule? In the glomerular filtrate? It is mostly water, salts, minerals, it can be sugar may be present. So all these will be reabsorbed. back into the blood. So that is called selective reabsorption. So what is selective reabsorption? Reabsorption of water, salt, sugar and other minerals present in the glomerular filtrate back into the blood. So by the time the glomerular filtrate reaches the end of DCT, it gets concentrated, and that concentrated glomerular filtrate contains water and mostly urea. That is what is known as urine. So that urine will be now secreted into the collecting tubule. So we can call it tubular secretion. Tubular secretion. So the urine, when it is, so the glomerular filtrate has now taken the form of urine, and this urine is disposed of through the collecting tubule, and the blood regains most of its components and it flows back without the urea in it. Now I'll show you how these nephrons are located in the kidney. So suppose this is the kidney. So we have the collecting tubules which are present like this, radially arranged collecting tubules. And in the collecting tubules, each collecting tubule have lots of nephrons connected with it. And the nephron will be supplied with blood vessels, nephron will form the urine and it will be secreted into the tubular part. The urine will come through the collecting tubules and the urine will move out through the ureter and it will get stored in the urinary bladder. So this is how urine formation takes place inside a nephron. Thank you.